So in the previous session, we all started with our biochemistry of proteins. So in the last session, we have seen biochemistry of amino acids. In the last session also, we have seen some points of your proteins, but I would like to revise it again. I have added some few slides, more slides to last presentation. So I would like to revise it. We were discussing about the amino acids. We have seen the amino acids, their classification, their properties. Now you know that proteins are nothing but the polymer of amino acids. Proteins are nothing but the highly rich nitrogen containing organic molecules which are a polymer of L alpha amino acids. They are the polymer of L alpha amino acids. And if you see 16 to 20% of your body weight, dry body weight, so that will comprise a protein. आपका जो बॉडी वेट है अगर जला उसका 20% जो ड्राई बॉडी वेट, उसका 20% जो है वो प्रोटीन होता है. Proteins are formed from the amino acids by formation of peptide bond. Where two amino acids will react with the amino group of one amino acid and carboxy group of another amino acid. When they will react together to form a peptide bond, CONH bond. Okay. Now for a one peptide bond you need two amino acids. For a two peptide bond you need three amino acids. Likewise that for a tripeptide you need a two double bonds. Sorry, for a tripeptide you need two peptide bonds and three amino acids, right? Likewise that. Okay. So if more than 50 amino acids are there, it is known as a simple peptide. It is known as a simple peptide. So proteins are nothing but the polymer of L alpha amino acids, right? For a formation of one peptide bond you need two amino acids. For two peptide you need three amino acids. For 4 peptide bond you need a 5 amino acids. Likewise decapeptide will have, right, decapeptide will have peptide bonds and 11 amino acids, right? So amino acid sequence in the protein can be recognized or can be identified, can be determined by reagents. So the name of reagent is nothing but the Sagnus reagent. And what is the reagent? 1-fluoro 2,4 dinitrobenzene. So this reagent particularly react with the amino terminus of your amino acid. NH2 group जो होता है, that is known as amino terminus. In COOH group, okay, that is known as the carboxy terminus. So all these these reagents, Edman reagent, then your Sagnus reagent, what they will do, they will react with the amino terminus of the protein, amino terminus of the amino acid present in the protein, and that can analyze the amino acid sequence in the protein. Edmonds reagent is there. So reagent का नाम याद रखना है. Edmonds reagent is nothing but the phenyl isothiocyanate. Sagnus reagent is nothing but the 1-fluoro 2,4 dinitrobenzene. Question इसके ऊपर आ चुके हैं already. Now next, primary structure of protein. Now I would like to revise in a very fast because we have already seen in the last session. So primary structure of protein means what? It's simply a peptide chain is there. Okay. So simply amino acid sequence is there. There is a peptide bond is involved in this. So CONH bond. This is known as a peptide bond. Okay. Linear structure, primary structure में linear protein का structure होता है, linear structure, and there is a only sequence of amino acid. There is only a sequence of amino acid, and there is a peptide bond. Okay. Now if you want to read a peptide, so you have to read from N terminus. You have to start reading the peptide in terms of amino acid from N terminus to C terminus. Okay. This is also case. This is also case when there is a synthesis of protein. तो प्रोटीन सिंथेसाइज होता है आपके बॉडी में. So also synthesis will start from N terminal and it will go up to the C terminal. Okay. N means NH2 group of amino acid and C means carboxyl group of amino. Now two amino acid will form one peptide bond and all that already seen. Now in the linear, that is known as the primary structure of protein. Secondary structure means what? Secondary structure of protein means twisting of your primary structure. एक प्राइमरी स्ट्रक्चर है आपका लीनियर पॉलीपेप्टाइड है, लीनियर पेप्टाइड है, वो अगर ट्विस्ट होता है या टर्न होता है, जैसे ट्विस्ट हो गया, टर्न हो गया, सिंगल ही है, ट्विस्ट एंड टर्न, बिकॉज ऑफ दैट देयर इज अ increased interaction. Now in the secondary structure, that is known as twisting, it is a structure by twisting and turning. Now it is proposed by Pauling and Corey. There was two scientists, Pauling and Corey. So they proposed the secondary structure of protein. They proposed a secondary structure of protein, and there are also two types they have mentioned. So Pauling and Corey. Pauling and Corey. This here is written only. Pauling and Corey, they have depicted or they have given the secondary structure of protein, and in that in that you have alpha helical structure of protein and beta plated sheet. In that you have alpha helical structure and beta plated sheet. Okay. Now in this structure, in addition to the CONH bond, in addition to the this peptide bond, so additional bonds are there. Hydrogen bonding will be there. Hydrogen bonding will be there. Hydrogen bondings of different amino acid between two peptide chain or simply same peptide which is being twisted or turned. एक अगर एक ही पेप्टाइड है एक साइड से टर्न होके फिर से बैक साइड में गया ओके ट्विस्टेड बैक if peptide is twisted back so there might be interaction of NH okay there might be interaction of H and carboxy group. So there is a hydrogen bonding over there. So in the secondary structure, you have to remember secondary structures they are stabilized by hydrogen bonding. So in that you have alpha helical structure and beta plated sheet. Okay. So in the alpha helical structure, we will see in the each turn of alpha helical structure there is a 3.6 amino acid which are at a distance of 0.54 nanometer and distance between two amino acid. So it is a 1.5 nanometer. Now 1.5 nanometer. There are different helix. Right-handed helix is there. Left-handed helix is also there. Right-handed helix is more stable than left-handed helix. The amino acid like proline, amino acid like proline or that is amino acid, acidic amino acids like aspartic acid, glutamic acid. एसिडिक अमीनो एसिड जो होता है प्रोलीन जो होता है बेसिक अमीनो एसिड जो होता है बेसिक है ना LH हिस्टिडीन हिस्टिडीन aspartame and lysine that also disturb the alpha helical structure of protein. Now beta plated sheet is also one of the structure beta plated sheet. Now they are proposed by Pauling and Corey. Remember, so it is known as secondary structure of protein. We will see the difference between alpha helical structure and beta plated sheet. I have summarized that difference. Now I have included that slide also. Now in the tertiary structure of protein, so all these are nothing but the hierarchy level of your protein structure of protein कि प्रोटीन का स्ट्रक्चर कैसे होगा प्राइमरी हो सकता है सेकेंडरी हो सकता है टर्शियरी हो सकता है क्वाटरनरी हो सकता है प्राइमरी में सिर्फ CONH बॉन्ड रहता है और लीनियर स्ट्रक्चर होता है सेकेंडरी में क्या होता है कि देयर इज अ इंटरेक्शन बिटवीन देयर इज इंटरेक्शन बिटवीन हाइड्रोजन आयन एंड carbonyl ion of two amino acid of two peptide or two amino acid of same peptide which is twisted back right so because of that interaction there is hydrogen interaction and because of that twisting and turning is there and that leads to protein secondary structure of protein now tertiary structure so it is nothing but the three dimensional arrangement of protein okay three dimensional arrangement of protein okay so there may be two or more polypeptide which are arranged in a three dimensional. Now in this case hydrogen bonds are there disulfide bonds are there ionic bonds are there okay ionic bonds are there hydrogen bonds are there disulfide bonds are there van der Waals forces are there and hydrophobic interactions are there. Now they may ask you a question like this which okay so secondary structure of protein is stabilized by following type of bond. So hydrogen bond. Primary structure there is only having peptide bond. Okay. And in the tertiary structure all hydrogen bond, so disulfide bond, ionic bond, van der Waals forces, hydrophobic interaction. Now in case of quaternary structure, so lot of polypeptide chains they will come together and there is a interpeptide hydrogen bond over here. Two or more polypeptide they are held together by hydrogen bond, disulfide bond, all bonds including tertiary structure. Okay. Now example of this quaternary structure is the hemoglobin and you can see okay प्राइमरी सेकेंडरी स्ट्रक्चर और यू कैन सी टर्शियरी स्ट्रक्चर का एग्जांपल होता है आपका इंसुलिन सो इंसुलिन इज अ स्ट्रक्चर टर्शियरी स्ट्रक्चर नाउ मेथड्स टू मेजर द प्रोटीन स्ट्रक्चर secondary and tertiary structure of protein can be analyzed by x-ray crystallography okay and that is a most commonly used method am I audible to you okay, most commonly used method for secondary and tertiary structure of protein. For determination of secondary and tertiary structure of protein, that is a X-ray crystallography, which is the most common method. इसके ऊपर क्वेश्चन ऑलरेडी एक बार आ चुका है. Most common method for determination of structure of protein, secondary and tertiary structure of protein, that is a X-ray crystallography. Now again, NMR, nuclear magnetic resonance, will also give you idea about the structural and functional information of your protein. But remember this X-ray crystallography, which is used for the determination of secondary and tertiary structure most commonly. Transamination, deamination, these are the two reactions mostly. They are in a metabolism of your proteins and amino acids. Transamination means what? Transamination means only transfer of amino group from one amino acid to the another keto acid to form a new amino acid. Okay? Now this is how there is a formation of peptide bond, not transamination. So how there is a formation of peptide bond? When two amino acids will come together. So amino group of one amino acid and carboxylic group of another amino acid, when they will react to form a CONH bond. CONH bond. This is known as a peptide bond. Okay? Peptide bond. A dipeptide will have one peptide bond and three amino acid. Sorry, a dipeptide will have one peptide bond and two amino acid. Okay, a monopeptide. Okay, dipeptide. Okay, dipeptide. इसको dipeptide बोलते हैं. Dipeptide means having two amino acids and one peptide bond. को बहुत सारे बुक्स में इसके ऊपर कंफ्यूजन है कि dipeptide means two peptide bond or one peptide bond. Now comparison between alpha helix and beta plate sheet we have already studied seen. So alpha helical structure, which is a okay. In this case, the peptide bond is a backbone. Peptide bond is backbone, and amino acids they are extended peripherally. Amino acids are extended peripherally. Now hydrogen bonds are parallel to the polypeptide backbone. Hydrogen bonds are parallel to the polypeptide backbone. You can see जो बीच में line draw किया है, that is a backbone. Now this is hydrogen bond, which is a parallel to the backbone, which is parallel to the peptide backbone. Okay, so hydrogen bonds in the alpha helical structure, which is a parallel to the backbone. Okay, now axial distance between amino acid is 1.5 angstrom. Okay. Now in case of beta plated sheet, so hydrogen bonds are perpendicular to the backbone of your peptide. So you can see over here. So what is backbone? Backbone ये यहाँ पे CONH bond. This is the backbone. Now in this case, hydrogen bondings they are perpendicular. Just remember these two things. Okay. One can constitute a polypeptide chain. One or more constitute the polypeptide chain. मतलब in case of alpha helix structure, so most of the time one peptide chain is there and which will compose a alpha helix. Now in case of beta plated sheet, it might be one peptide chain or it may be one or more peptide chains which will form a beta plated sheet. Right? So again you can see the comparison between the same thing. So we have already seen. So in case of alpha helix and beta plated sheet. Now you can see the difference between beta plated sheet and alpha helical structure of protein. So it was by Pauling and Corey. Now alpha helix structure क्या होता है? Alpha helical structure, that is nothing but a tightly packed or coiled structure. So amino acid chains which are extending outward from the central axis, and alpha helical structure it is stabilized by hydrogen bonding between hydrogen of peptide N terminus and oxygen of peptide C carboxyl terminus. So alpha helical structure they are stabilized by hydrogen bonding. Now all peptide bonds take parts in the hydrogen bonding except first and last. In case of alpha helix है ना, जो peptide है, all peptide bonds, okay, जो peptide है उसमें के जितने भी bonds है, they will take a part in the hydrogen bonding except first and last peptide bond. Alpha helix is a stable conformation and it is formed by spontaneously with the lowest energy. That's why it is stable. It is a right-handed alpha helix more stable than left-handed alpha helix. A few amino acids they will disturb or they will destroy the structure of your alpha helix. So proline, acidic amino acid and basic amino acid. एसिडिक में कौन होता है, एस्पार्टिक एसिड, ग्लूटामिक एसिड। इसमें क्या है बेसिक में, इसमें आता है एस्पार्टामाइन है ना, इसमें एलएच है ना, हिस्टिडीन, लाइसिन एंड aspartame. Now beta plated sheet you can see over here. It is composed of two or more segment of fully extended peptide chain. यहाँ पे दो या फिर एक या दो peptide chain होगा. Hydrogen bonds are found between neighboring segment of peptide chain. Now parallel and anti-parallel beta plated sheet. There are the two types of beta plated sheet. Hydrogen bond between two peptide chain or hydrogen bond between same peptide chain which is folding back. So that occurs in the beta plated sheet. So this was a new slide to you. I hope you are getting. Am I audible to you? We have seen yesterday this. Yes or no? Am I audible? So we have seen yesterday the primary structure of insulin. So insulin is composed of a two chain. Okay, native insulin. Okay. Now insulin is synthesized from pre-proinsulin. Pre-proinsulin. Right. Now pre-proinsulin has a 110 amino acid. प्री-प्रो इंसुलिन होता है उसमें 110 अमीनो एसिड 110 110 amino acids pre-pro insulin. So after pre-pro insulin it is a broken down by proteolytic enzymes. Now it is converted into pro insulin. What is pro insulin? So this is a pro insulin. So 86 amino acids are there in a pro insulin. So pro insulin will have three chains. Okay. So ये होता है A chain, ये होता है B chain, and ये होता है C chain. Now from the pro insulin again by the proteolytic enzymes will act at this side. यहाँ पे और यहाँ से कट करेगा कौन, enzymes, proteolytic enzyme. Now there is a removal of this C chain, that is known as a connecting peptide, and you will get your active insulin. Active insulin is nothing but having 51 amino acids. एक्टिव इंसुलिन में कितना अमीनो एसिड होता है 51. क्वेश्चन आता है इसमें A chain will have how many number of amino acids? So A chain will have 21 number of amino acids, 21, and B chain will have 30 amino acids. B chain will have 30 amino acids, right? You have to remember. Now we'll see the next part. Determination of primary structure of protein. So proteins are either hydrolyzed by acid or enzyme completely to liberate amino acids, which are then sequentially quantized or estimated. Right? There is one mixture, pronase. Pronase is a mixture of protein that can completely hydrolyze a protein. Then amino acids are separated by chromatography and then they are estimated. Right? They are estimated. Degradation of protein or polypeptide into smaller fragment. So you can also break down the protein into smaller fragment because it is treated with the urea or guanidine hydrochloride to liberate a polypeptide. By now this guanidine hydrochloride or urea एक कैसे act होता है? They disturb the non-covalent bonds. And causes the dissociation of protein. Now dancing chloride is also there, one reagent which will binds to N terminal of amino acid, and when it is treated it reacts with the protein and it is used to identify number of polypeptides. Polypeptides are broken down into small fragment by enzymatic or chemical method. Now उसके chemical method और enzymatic method होते हैं. Now by using Sagnol's reagent also, Edmonds reagent also, and there is one automatic machine known as a sequenator which will determine the sequence in the amino acid sequence of protein. Types of bond present in the protein. So there are two type of bond. एक होता है covalent bond, है ना, covalent bond होता है, दूसरा होता है non covalent bond. तो covalent bond कौन सा होता है भाई? Covalent bond present in the protein is nothing but the simply a peptide bond. So CONH bond, peptide bond is a covalent bond, and then second is nothing but the disulfide bond. Now disulfide bond which is formed between two sulfur containing amino acid, है ना, अगर पेप्टाइड में दो अमीनो एसिड सल्फर कंटेनिंग अमीनो एसिड उसमें डाइसल्फाइड बॉन्ड बनेगा, राइट. So it is a covalent bond and this peptide bond is also covalent bond. Now non-covalent bond present in the protein structure, that is hydrogen bonds, hydrophobic bonds, interval interaction, electrostatic bonds. These are the types of bond present in the protein structure. ये आपको मालूम होना चाहिए, इसके ऊपर क्वेश्चन आ सकता है. What are the covalent bonds present in the protein structure? What are the non-covalent bonds present in the protein structure? Okay. Then Properties of protein we have seen. Molecular, they will form a colloidal solution with the water. Molecular weight will range 4000 to 44 okay 4 lakh 40,000 Dalton. इंसुलिन का होता है 500 sorry 5700 Dalton. Myoglobin 1070,000 Dalton. Then hemoglobin 64.45 Dalton. Then immune albumin, you know that इसके ऊपर क्वेश्चन भी आ चुका है एक बार. So the range is nothing but the 64 to 69,000 Dalton, and the answer was given 65,000 Dalton in GPAT 2021. There was a question. Now protein का shape कैसा होता है? Protein का shape, it may be either oval shape, it may be either fibrous shape, it may be either इंसुलिन इज अ ग्लोबुलर शेप. इंसुलिन का जो शेप होता है वो ग्लोबुलर होता है. अल्बुमिन इज अ ओवल शेप एंड फाइब्रिनोजन इज अ fibrous shape or elongated shape. So shapes of protein, right? Then isolytic pH means what? The pH at which amino acid will exist as a zwitter ion or dipolar ion, है ना. तो अमीनो एसिड में अमीनो ग्रुप होता है NH3 प्लस प्लस NH2 प्लस प्लस उसमें COO माइनस ग्रुप होता है. So amino acid has a both anionic group or cationic group. So if you place amino acid in the acid environment, basic group will be protonated. So basic group will be protonated. If you keep amino acid in the basic, so acidic group will be protonated. So it acts as a buffer. Now there is a particular pH at which the amino acid will exist as a dipolar ion. Amino acid will exist as a zwitter ion. Now that is known as a isolytic pH of your amino acid. Okay, and at the isolytic pH, it has a bipolar ion. It is a neutral. Okay, at the isolytic pH, amino acid will carry no net charge because okay, and in the similar way, proteins are nothing but the polymer of amino acid. So protein also has isolytic pH. What is isolytic pH of protein? Same thing. But at the isolytic pH, protein has a minimum solubility. Isolytic pH के टाइम पे प्रोटीन को क्या होता है? Minimum solubility होता है, right? So you can see isolytic pH of pepsin is a 1.1, casein 4.6. So you can see if you take a milk, अगर मिल्क दूध अपने रख दिया तो दूध में क्या होता है? एक दिन में फट जाता है. वो क्यों फटता है? Because bacteria will get in that milk. Contamination of milk will be there by bacteria, and bacteria will cause release of some chemicals which will decrease the pH of your milk. Now if pH of milk comes to 4.6, what happens? As I told you, at this pH in the protein, the pH at the isolytic pH, protein has a least solubility, and protein also coagulates. So what happens at the 4.6? If pH of milk comes to 4.6, and what you are doing, अगर आपको दूध को फाड़ना है तो आप क्या करते हो? फिर उसमें नींबू डालते हो. नींबू डालना मतलब क्या होता है? नींबू में क्या होता है? एसिड होता है ना, साइट्रिक एसिड. सो दैट साइट्रिक एसिड आल्सो डिक्रीजेस द पीएच ऑफ योर मिल्क. जैसे बाकी बैक्टीरिया करते हैं वैसे साइट्रिक एसिड भी करता है ना. जब pH 4.6 पे आता है, so what happens? There is a coagulation of casein, and what we call it is a curd, है ना. वो दूध फट जाता है, कर्ड बनता है, because milk is nothing but the kind of emulsion. Now albumin has a isolytic pH 4.7. Urease enzyme will have isolytic pH 5, and hemoglobin has a 6.7. So isolytic pH of protein. Okay, so at this isolytic pH, protein will have a minimum solubility, and they used to get coagulate at the isolytic pH. आपको ये याद रखना है point. Now precipitation of protein. Protein can be precipitated by a different way. Dehydration or neutralization of polar groups in the protein at acidic pH. Then precipitation of protein by salting out. If addition of neutral salt is there to the quaternary structure of protein. By adding or dehydration by using neutral salts such as ammonium sulfate and sodium sulfate. Again, protein can be precipitated by heavy metals like lead, mercury, zinc, cadmium. Okay, they can precipitate the protein. Or even you can simply precipitate the protein at a isolytic point. Just decrease or just set the protein solution at a isolytic point. So there is a precipitation of protein. Denaturation of protein means what? So loss of native structure of protein. जो native structure है, नॉर्मल स्ट्रक्चर है प्रोटीन का, उसका स्ट्रक्चर का लॉस हो गया, मतलब उसको बोलते हैं अपन डीनेचुरेशन ऑफ प्रोटीन। तो इसके ऊपर भी कभी क्वेश्चन होता है। तो डीनेचुरेशन ऑफ प्रोटीन इज नेटिव लॉस ऑफ नेटिव स्ट्रक्चर ऑफ प्रोटीन। नाउ इट मे बी फिजिकल और इट मे बी बाय फिजिकल मेथड और केमिकल मेथड। फिजिकल मेथड में क्या है कि विगरस शेकिंग होता है, अपन देखेंगे, और वो भी स्लाइड है। फिजिकल मेथड में विगरस शेकिंग होता है, हीट होता है ना, हीट विगरस शेकिंग। केमिकल मेथड में एसिड अल्कली वगैरह है ना। सो नेटिव स्ट्रक्चर ऑफ प्रोटीन, there is a denaturation of protein. And physical method, okay, loss of physical structure. Okay. In case of denaturation, there is a loss of physical structure, but primary structure जो होता है that remains intact. But what is primary structure? Primary structure of protein is nothing but the CONH bond. So that remains intact. Primary structure or remains intact. Loss of biological activity, that can result in the loss of biological activity. Denaturation of protein can result in the insolubility. जो प्रोटीन सोल्यूबल है, डीनेचुरेशन होने के बाद में इनसोल्यूबल होगा, राइट। सो द वेरी फेमस एग्जांपल ऑफ दिस, ओके, डीनेचुरेशन ऑफ प्रोटीन रिजल्ट इन द इनसोल्युबिलिटी। जैसे फॉर एग्जांपल आपके पास में अंडा है ना, मुर्गे का अंडा, तो अपने पास एक सवाल भी है वैसा, मुर्गी आई पहली या अंडा पहले आया, मुर्गी पहले आई, अभी तक वो कोई सवाल कोई सुलझा नहीं सका, है ना। बट अगर अंडे को आपने बॉईल किया वाटर में, तो क्या होता है कि उसमें जो लिक्विड प्रोटीन होता है दैट denaturates and that coagulates. So denaturation of protein result in the insolubility. Okay? Increase in the viscosity. Okay, again. Am I audible to you? Are you getting or not? आवाज येते का? आवाज येत आहे का? Denaturation also result in the increase in the number of ionizable groups, sulfidal groups, and denaturation can result in the protein which can be easily digested. जो नेचुरल नेटिव प्रोटीन है दैट इज नॉट इजी टू डाइजेस्ट, बट डीनेचुरड प्रोटीन इज इजी टू डाइजेस्ट, है ना। आपने देखा होगा किसी ने कच्चा मटन खाया गया, है ना, गया वहां पे मटन की दुकान पे मटन ले लिया और वहीं पे ही खाने लगा, नहीं, क्यों, वो घर पे आता है, उसको पकाता है और बाद में खाता है। पकाना मतलब क्या, देयर इज अ डीनेचुरेशन ऑफ प्रोटीन। सो पकाना मीन्स नॉट, देयर इज अ डीनेचुरेशन, हीटिंग that causes denaturation, and that denatured protein is easy to digest, easy to digest. Denaturation is usually irreversible. Denaturation जो होता है normally irreversible होता है, and careful degradation sometimes is a reversible denaturation. Denaturation is normally irreversible, but careful degradation of protein that can result in the renaturation. Okay. Careful degradation sometimes is a reversible and it is known as a renaturation. Renaturation. Now, heat coagulation test is the most commonly used to detect the presence of albumin in the urine. आपकी यूरिन में अल्बुमिन है कि नहीं है ना, चेक करने के लिए सिंपल है, यूरिन को टेस्ट ट्यूब में ले लो, उसको थोड़ा हीट करो बर्नर के ऊपर। इफ देयर इज अ turbidity, that means there is a albumin in the urine. Now, if normally also if in your albumin is there, that means you are suffering from either nephritis or you are having some kind of infection. तो उसका इन्फ्लुएंस से होता है if protein is found in the urine. Now next part we'll see. So again, level up organization of protein. So level up or protein organization. Primary structure of protein, it is nothing but the linear structure. Simply peptide bond is there. Sequence of chain of amino acid is there, right? Now in the secondary structure, so which occurs when sequence of amino acid are linked by hydrogen bond. There is a twisting and turning of प्राइमरी स्ट्रक्चर। प्राइमरी स्ट्रक्चर का ट्विस्टिंग एंड टर्निंग, तो इसके बीच में हाइड्रोजन बॉन्ड रहेगा, है ना, ये लूप हेलिक्स के बीच में। देयर आर टू मॉडल्स प्रपोज्ड, अल्फा हेलिकल स्ट्रक्चर एंड बीटा प्लेटेड शीट, राइट। नाउ टर्शियरी स्ट्रक्चर ऑफ प्रोटीन, if there is a mixing between different kind of peptide chains, it may be either alpha helix or beta plated sheet. So occurs when certain attractions are present between alpha helical structure and plated sheets. So here is one alpha helical structure and beta plated sheet. That is another tertiary structure of protein. Now quaternary structure is a protein is consisting of more than one amino acid side chain. अमीनो एसिड चेन दो तीन हो गए तो उसमें attractions like hydrogen bonding, and because of that quaternary structure of protein is there. Quaternary structure of protein is there, right. Classification of proteins are classified into these three major classes: simple proteins, then conjugated proteins, and derived proteins. Now what do you mean by simple proteins? Simple proteins, there are further divided into two types: globular proteins. So they are having globule like shape, globular proteins. They are soluble in the water. Okay. Now in that you are going to have albumin, globulins, okay, glutenins, prolamines, histones, globulins, protamine and lecithins, protamine and lectins. Then scleroprotein. Scleroprotein, or it is known as a structural protein. Scleroprotein को ही structural protein बोलते हैं. In that you are going to have collagen, elastin, and keratins. Collagen is found in the connective tissue. Elastin is also found in the collagen. If you heat a collagen with the water, or dilute acid, so collagen will give you gelatin. Collagen जो होता है वो आपको gelatin देगा. Elastin and keratin. Keratin is found in the hairs, nails, and horns of animals. और मतलब हॉर्न मतलब एनिमल के पैर के नीचे जो होता है उसका नाखून बड़ा वाला हॉर्न्स, या फिर आपका सींग जो होता है बैल का सींग, उसको भी हॉर्न बोलते हैं. Conjugated proteins. Conjugated protein means these are having proteins with some additional group known as a prosthetic group. Conjugated protein मतलब क्या? Protein plus additional group known as a prosthetic group. Now in this case nucleoprotein. Now nucleoprotein में कौन सा prosthetic group रहेगा? Obviously DNA RNA वगैरह ये रहेगा, protein plus DNA RNA. Then glycoprotein में क्या है? Glyco मतलब sugar, a complex of sugar plus protein. So in this case carbohydrate is a prosthetic group. In that only mucoprotein is the mucoproteins. So when carbohydrate content is okay, when carbohydrate contains more than 4%, when carbohydrate content is more than 4%, then it is known as a mucoprotein. So lipoprotein means macromolecular complexes of protein with the lipids. Phosphoprotein means यहाँ phosphoric acid क्या रहेगा? Prosthetic group. Chromoproteins, chromo मतलब क्या? Chromo is for color, है ना? Protein मतलब protein. Chromoprotein मतलब color protein. क्यों होता है भाई color? जैसे आपका hemoglobin है, hemoglobin का color क्या होता है? Red, है ना? So chromoprotein. Okay. Then metalloprotein, complex of metal with the protein. Then derived protein, they are nothing but the hydrolytic product of your simple and conjugated protein. अगर जो पहले दो है conjugated simple, उसका हाइड्रोलिसिस करेंगे, उसका है ना डिग्रेडेशन करेंगे, उसका कोएगुलेशन करेंगे, उसका डीनेचुरेशन करेंगे. So they are the degraded, denatured, coagulated product of your simple and complex protein. Simple and conjugated protein. Conjugated protein also known as complex protein. Okay. Now in this primary is there, primary coagulated. So coagulated proteins, proteins, and metaproteins. In secondary में आता है, secondary derived protein में आता है primary derived protein, secondary derived protein, proteose, peptones, polypeptide, and peptide. This is the classification. We will see in a short the example of say individual का example देखेंगे अपन. Now you can see classification of protein over here again. So this is a new slide I have added. कल ही सुबह निकाले. Now classification of protein, functional classification. So based upon the function of protein they are classified. So functional classification. Okay. So with the class sign example. Now इसमें आता है structural protein. So they are the found in the structure. Like keratin is there, collagen is there. Keratin and collagen. They are the structural protein. Some protein will act as an enzyme. So for example like pepsin and hexokinase. Pepsin is a proteolytic enzyme. Hexokinase is the enzyme used in the enzyme has a role in the carbohydrate metabolism. Yeah. So proteins as a transport protein. इसमें आता है hemoglobin and albumin. So what is the role of hemoglobin? To transport the oxygen, some amount of carbon dioxide. Albumin also involved in the transportation of your vitamins, amino acids, and some drugs also bind to the albumin. Then some proteins act as a hormone. Example insulin and growth hormone. Some proteins they are contractile protein. They are involved in the muscle contraction. यहाँ पे actin and myosin क्या होता है? Responsible for the muscle contraction. Some proteins they act as a storage protein. For example, O-albumin and glutenin. O-albumin and glutenin. O-albumin कहाँ पे होता है? ये eggs में होता है. ग्लूटेलिन का होता है, इट इज फाउंड इन द ओ एल्बुमिन, मतलब ओम जो होता है एग्स, ग्लूटेलिन अगेन है ना, इट इज फाउंड इन द wheat. Now again, genetic protein like nucleoproteins, some proteins they are having function in the defense mechanism. For example, snake venom protein and hemoglobin. Some proteins they act as a receptor, say for example hormonal receptor. These are the structural classification of protein. Are you getting or not? समझ रहा है आप नहीं? Yes or no? Are you getting or not? Now again, classification of protein based upon the chemical nature and solubility. So based upon that, they are classified into three types: simple proteins, conjugated proteins, and derivative proteins. We have seen that. Okay. Yeah. Now again, okay. Simple means simply amino acid chain will be there. Simple protein matlab amino acid chain will be there. Bound by peptide bond simply. Conjugated protein conjugated protein matlab amino acid residue, okay, that is peptide chain plus prosthetic group, that is a non-protein group or non-protein molecule. So derivative protein in that natural or degraded product of simple and conjugated proteins. Now denaturation मतलब क्या होता है प्रोटीन का? denaturation of protein मतलब loss of native structure of protein, loss of native structure of protein. Now how you can denaturate the protein? There are different denaturing agent. In the physical you have heat, है ना, आपने heat apply किया, protein का denaturation हो गया. So that is the mechanism involved in the again dry heat sterilization. You see dry heat sterilization में क्या होता है कि आप जो sterile करना है आपको hot air oven में रख देते हो, है ना. So 180 degrees Celsius for 1 hour, है ना, 3 hours, 2 hours. So what happens during that? So that heat causes the denaturation of bacterial proteins and enzyme. That's why bacteria will die. Right? So denaturation is the mechanism. Again, same mechanism in the uh, in the moist distillation. So in the dry distillation, denaturation as well as oxidative damage to the bacterial enzymes and DNA and protein. So that is the mechanism in the dry distillation. Now, in the moist distillation, principal mechanism is a coagulation of protein and denaturation. So you can denaturate the protein by using heat, X-rays, UV rays. Then vigorous shaking is also the vigorous shaking. Okay. Then chemical method is also there. Chemically, you can denaturate the protein by using chemicals. For example, by using acids, alkali, organic, solvent salts, heavy metals, urea, salicylic acid, detergents. So they can also denaturate the protein. देखो आप है ना, आपको बोलते हैं हैंड वॉश करो, आपको हैंड वॉश करो, है ना. करोनामध्ये काय सांगितलं की सरळ सरळ साधा तुम्ही हाथ 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 धो चला. कोरोना में क्या हुआ था कि बोला था कि हैंड वॉश करना बहुत ही इम्पोर्टेंट है ना. हैंड सैनिटाइजर ये वो यूज करने लगे लोग. पहले तो सिंपल सिंपल डिटर्जेंट जो होता है आपका सोप है घर में, सोप जो आपका है, सोप से भी हाथ वॉश किया आपने ना, तो उसमें भी वो जर्म्स वगैरह मर जाते हैं. क्यों? बिकॉज ऑफ डिटर्जेंट्स, because of the detergents. So it causes the denaturation of protein and bacteria virus will die. Okay, that's why. लेकिन बिजनेस करना था लोगों को, लोगों ने क्या कर दिया, अल्कोहल बेस्ड सैनिटाइजर. then coagulation of protein is irreversible denaturation of protein at okay lowest temperature, at which okay lowest temperature at the isolated pH leads to coagulation, है ना. coagulation और एक तरीके से होता है lowest temperature. When there is a lowest temperature at a isolated pH of that protein, there is a coagulation of protein. Then again flocculation is there. flocculation मतलब क्या? precipitation of protein at a isolated pH. example for example casein. casein क्या है? milk protein होता है. casein is nothing but the milk protein. casein जो है it is a milk protein and it is precipitated from the milk by adjusting the isolated pH at a 6 at a 4.6, and it is a reversible and it can be made irreversible by application of it. Yeah. So flocculation is a reversible phenomenon. Reversible. but by application of heat to the floccules, then it becomes irreversible. So about the denaturation of protein. तो ये सब additional point था, कल नहीं था. Now again you can see, we'll see the again proteins, different proteins. I hope I am audible to you, and are you able to see the screen? Are you getting? Now again you can see simple proteins. Simple proteins, types of protein, simple protein. So simple में आता है पहला globular protein. Globular protein, because they are known as globular protein. Why? Because they has a oval shape or a spherical shape. They are soluble in the water and organic solvent, and they are also digestible. You can easily digest them. Now in that you are albumin. Albumin क्या है? Albumin has a different type. Serum albumin is there. O albumin. O albumin कहाँ पे होता है? Eggs में होता है. Lactalbumin. Lactalbumin कहाँ होता है? Milk में होता है. They are the water soluble and they are coagulated by heat. Albumin. ग्लोबुलिन जो होते हैं, ग्लोबुलिन फॉर एग्जांपल सीरम ग्लोबुलिन होता है ना. देन विटेलिन होता है, एग yolk जो होता है, येलो एग yolk, विटेलिन. देन सोल्यूबल, these globulins are soluble in the salt and neutral solutions. नेक्स्ट होता है ग्लूटिलिन्स. ग्लूटेलिन्स. सो ग्लूटेलिन इन द प्रोटीन. देन ओराइजिन. ओराइजिन कहाँ से आता है? ओराइजिन आता है राइस से. इट इज अ प्रोटीन फ्रॉम राइस. एंड ओराइजिन फ्रॉम glutenin from wheat. Glutenin is found in the wheat. ये जो glutenin होता है ये wheat में होता है. ये origin जो होता है वो rice में होता है. So glutenin origin from the wheat and rice respectively. They are soluble in the dilute acid and alkalis, and they are mostly found in the plants. Next, the prolamins. Prolamins example is a gliadin and zein. Gliadin is from a wheat and zein is from maize. Zein क्या है? Zein is also entry coating agent. Zein जो है it is a polysaccharide and it is entry coating agent. ना? It is a complex of protein plus saccharide. They are soluble in the 70% alcohol. Next होता है stones. So stone का example है thymus stone. They are the strong basic protein soluble in the water and dilute acids but insoluble in the alkali. Lecithin is a carbohydrate binding protein. Example concanavaline. Concanavaline. Okay, concanavaline and concanavaline A and A-glutinins. They are the lecithin. Lectins है या lectins है. Lectins. नेक्स्ट होता है आपको फाइब्रस प्रोटीन. फाइब्रस प्रोटीन, फाइबर, they are looking like a fiber, that's why they are known as fibrous protein. They are insoluble in the water. They are resistant for the digestion. एग्जाम्पल क्या है इसका? एल्यूमिनोइड्स एंड स्क्लेरोप्रोटीन. उसमें एक है पहला कोलेजन. कोलेजन इज प्रेजेंट इन द कनेक्टिव टिश्यू प्रोटीन्स विदाउट ट्रिप्टोफैन अमीनो एसिड. कोलेजन में ट्रिप्टोफैन अमीनो एसिड नहीं होता है. Now boiling of collagen with the water or dilute acids will give you gelatin. Gelatin is a digestible. Collagen is not digestible, but the the, the hydrolysis product of collagen is gelatin, and which is a digestible. Now elastin का आप में होता है आपका. It is found in the elastic tissue like tendons and arteries. Keratin आपका hair में पाया जाता है. Keratin is found in the hairs, human hair. So around 14% of cysteine amino acid is there in here. That's why cysteine is a sulfur containing amino acid. That's why your hair will have black color, right? Then also found in the nails and horns of animals. Keratin. Next होता है conjugated protein. So इसमें nucleoprotein होता है. Protein plus DNA plus RNA as a prosthetic group. Next होता है glycoprotein. Protein plus sugar. Next होता है phosphoprotein. Protein plus phosphoric acid. Example in case of casein. Chromoprotein मतलब क्या? Protein with the color. For example, chromoprotein का example याद रखना है आपको. Hemoglobin, cytochrome, the other color. Metalloprotein मतलब complex of protein with the metals. You know. Then derived protein जो होता है उसमें primary derived and secondary derived. Primary derived मतलब क्या? Coagulated protein, proteins and metaproteins. Derived, okay. Secondary derived protein मतलब breaking of peptide, okay. Okay, यहाँ पे there is a breaking of peptide bond. So simply, yes, anybody? Simply peptide bond, proteins and peptides are there, peptones are there. So this is a derived protein, right? Primary, secondary, tertiary. You are not getting. स्क्रीन में दिख रहा है क्या? मैं कितनी बार पूछा हूँ, स्क्रीन दिख रहा है, दिख रहा है. नेक्स्ट पार्ट विल नथिंग बट द overview of amino acid metabolism. So normally in your body around 100 grams of amino acids is stored. 100 gram of amino acid is stored in your body, right? It is known as the amino acid pool of your body, right? So as carbohydrates, they are stored in the form of glycogen. Lipids are stored in the form of fatty acids. Okay, there is no storage form of amino acid in the body, so that's why only 100 gram amino acids, which is known as the free amino acids, and they are known as a amino acid pool. तो 100 ग्राम अमीनो एसिड होता है आपका, इसमें से भी 50% अमीनो एसिड पूल इज कंप्राइज्ड ऑफ योर ग्लूटामेट ओनली, ग्लूटामेट एंड एस्पार्टेट, और बाकी इसेंशियल अमीनो एसिड का 10 to 20% रहता है, राइट? नाउ from where these amino acid comes, these amino acid coming from diet. So whatever protein you are taking daily, 75 gram protein you are taking, that is broken down and converted into amino acid. Now again, breakdown product of your own body's protein, that is also amino acid. Normally 300 400 milligram 300 400 grams of protein they are degraded daily. 300-400 ग्राम प्रोटीन जो है वो डेली डिग्रेड होता है बॉडी में से, और वो उसका क्या बनता है? अमीनो एसिड्स. Amino acids. Now from these amino acids, what you are going to get? These amino acids, they are utilized for first for production of keto acids. You know, amino acids say keto acids by deamination reaction. Deamination reaction अमोनिया रिलीज होगा. अमोनिया जो रिलीज हुआ है उसको और अमोनिया ग्रुप के साथ में कंबाइन करके बाकी एस्पार्टिक एसिड के साथ में एस्पार्टिक एसिड एस्पार्टामाइन के साथ में कंबाइन करके लास्ट में होता है यूरिया. नॉर्मली यू प्रोड्यूस अराउंड 50 to 30 ग्राम ऑफ यूरिया पर डे. इतना यूरिया आप बनाते हो और छोड़ देते हो जमीन में, राइट? नाउ amino acids again they can be utilized for the production of, they are utilized for the energy. So amino acid can be utilized for the production of energy, oxidation will be there. Okay. Again from amino acids you know that gluconeogenesis is there. So glycogenic amino acids, so those amino acids which are glycogenic in nature, they produce glucose. From amino acid only you are going to have synthesis of non-essential amino acids. From the amino acids only you are going to have synthesis of specialized product like जैसे आपका हीम होता है, प्यूरिन्स होता है, pyrimidines, creatinine, enzymes, hormones, they are also synthesized. From amino acid only you can synthesize the whatever required protein for your body. So proteins are synthesized. So this is the overall metabolism of amino acids, overall movement of amino acids, right? Now as I told you, every amino acid has its own metabolism. It is not possible to take all the metabolism. You can understand the protein metabolism can be understood by simply understanding the amino acid metabolism. Right? So metabolism of proteins and amino acids. When this is adult has around 100 grams of free amino acid pool, which is known as amino acid. Amino acid. So glutamate and glutamine, ये दो amino acids that will comprise around, that will constitute around comprise around 50% of amino acid pool, and remaining 10% will be of your essential amino acid. There is no storage form of amino acid. In case of carbohydrate you have glycogen, in case of lipids you have triglycerides. The storage form. Now 300 to 400 gram protein per day is required consistently. It is degraded and synthesized, which represent the body's protein turnover. There is one polypeptide known as a ubiquitin, which is a polypeptide which increases the degradation of protein. 30 to 50 gram of proteins must be supplemented in a diet per day to maintain the nitrogen balance of the body. Now metabolism जो होता है amino acid का, it undergo mostly by two reaction. So amino acid undergo reaction like transamination followed by deamination for the release of ammonia. For the release of ammonia. Now transamination is the process where there is only transfer of amino group. Transamination में सिर्फ transfer होता है amino group का from one amino acid to another keto acid to produce a new amino acid and new keto acid. That is a transamination. We already seen. Now carbon skeletal carbon skeleton remaining after the transamination, which can be converted into keto acid, or it can be utilized to generate the energy, or it can be used for the synthesis of glucose, formation of ketone body, non-essential amino acids. Transamination, that is a transfer of amino group from one amino acid to the another keto acid to form a new amino acid. The enzyme transaminase, or nowadays it is known as a amino transferase. एंजाइम का नाम क्या है? ट्रांसअमाइनेज. Nowadays it is known as a amino transferase, which is dependent on pyridoxal pyrophosphate. Pyridoxal phosphate is a coenzyme obtained from vitamin B6. Now transamination is a reversible. It is a reversible. Transamination is important for production of non-essential amino acids, energy. Glutamate is the only amino acid that will undergo oxidative deamination. Now transamination and deamination, though we are studying separately, transamination reaction आपको separate लग रहा है, deamination reaction आपको separate लग रहा है, but mostly in the body they occurs at a time. Whenever there is a transamination, somewhere there is a deamination. So glutamate deamination में दो टाइप का होता है, oxidative deamination and non-oxidative deamination. So oxidative deamination means only glutamate that can undergo oxidative deamination, and all other part of your body can deaminate the drug, deaminate the amino acid except glutamate. Okay? Because glutamate undergo oxidative deamination and that is done in the liver. Okay? Ping pong bye model is there for transamination reaction, which is proposed by Snell and Brustin. स्पेलिंग भी बराबर है, कल मैंने बोला था स्पेलिंग गलत है, लेकिन गलत नहीं है. Now deamination, so removal of amino group from amino acid, है ना, as ammonia for urea synthesis. Now there is never free ammonia in your body. फ्री अमोनिया आपके ब्लड में होता है नहीं. जैसे अमोनिया नहीं होता है, अमोनियम ग्रुप होता है, NH4 प्लस ग्रुप होता है. So that group immediately, if it is made, it is deaminated, immediately that group is taken and converted into carbamoyl phosphate in the liver, and immediately that urea cycle starts. Okay, so oxidative deamination is catalyzed by glutamate dehydrogenase, and oxidative deamination of amino acid by amino acid oxidase. This mostly occurs in the liver and kidney. दो mechanism होता है metabolism के लिए. Based upon that, I have already explained you urea cycle in the last lecture, but still, now non-oxidative deamination, again to liberate the ammonia without undergoing the oxidation. It is done by two enzymes, amino acid dehydratase enzyme to form a respective keto acid, and then amino acid dehydratase enzyme, dehydrase and desulfohydrase. Desulfohydrase मतलब क्या है ना. तो नॉन ऑक्सीडेटिव डिएमिनेशन कौन करता है? लीवर में होता है. किसका होता है? ग्लूटामेट का होता है ना, ग्लूटामेट डिहाइड्रोजनेस. नाउ नॉन ऑक्सीडेटिव डिएमिनेशन जो होता है, दैट इज डन बाय अमीनो एसिड हाइड्रेटेज एंजाइम टू प्रोड्यूस रिस्पेक्टिव कीटो एसिड, एंड आल्सो अमीनो एसिड डीसल्फाइड्रेस. डीसल्फाइड्रेस मतलब क्या? दोस हु आर सल्फर कंटेनिंग अमीनो एसिड. तो उसमें क्या है? सल्फर कंटेनिंग अमीनो एसिड में क्या होता है? तो उसमें एसएच ग्रुप भी रिमूव किया जाता है, डीसल्फाइड्रेस है. एसएच ग्रुप भी रिमूव किया जाएगा और अमोनियम NH4 ग्रुप भी रिमूव किया जाएगा to become a keto acid. The liberated ammonia in the transamination and deamination is transported and utilized to synthesize a new non-essential amino acids, purines, pyrimidines, and amino sugar. Based upon that, now disposal of ammonia. Okay, how ammonia is disposed of from your body? Based upon that, okay, so whether ammonia is excreted as it is, whether ammonia is converted into uric acid and then excreted, whether ammonia is converted into urea and then it is excreted. Based upon that, animals, they are classified into three types. Aminoteric means these animals, they remove the or dispose the ammonia in the surrounding water. Now uricotelic means what? These animals, they convert ammonia into uric acid, they remove the uric acid. तो इसमें रेप्टाइल्स और बर्ड में यूरिक एसिड होता है. यूरियोटेलिक मतलब क्या? अमोनिया is converted into urea in the mammalian cells, mammals and including man. अपन भी वही करते हैं, अमोनिया बना तो यूरिया में कन्वर्ट करके अपन छोड़ देते हैं नीचे वाले रास्ते, ठीक है. So ureotely, so okay, then toxicity of ammonia. Ammonia, if it is accumulated in the body, so it is increased level of ammonia is harmful to the brain. Ammonia, when it accumulates in the body, it result into slurring of speech, blurring of vision. It can cause tumors, you know, और अपने hepatic coma भी हो सकता है. May leads to coma, hepatic coma, finally death. That's why urea. So that's why urea is synthesized from ammonia. Now, urea cycle. So the urea cycle was proposed by Krebs. Okay, it is also known as a Krebs-Hanslet cycle. Okay, because it was proposed by the Hans Krebs, okay, and Kurt Hanslet. Hans Krebs and Kurt Hanslet, they were discovered this urea cycle, and that's why the urea cycle is known as a Krebs-Hanslet cycle. Okay. Okay, now urea is synthesized in the liver. Urea कहाँ पे synthesized होता है liver में. Urea has a two amino groups. Urea has a two amino group. So first is nothing but the which is coming from deamination, and another NH3 group is coming from aspartate. Now urea cycle or synthesis of urea is a 5-step reaction. इसको 5-step reaction होते हैं. Now first two steps that occurs in the mitochondria and all other steps that occurs in the cytosol. Now पहले तो enzyme जो है that is available in the mitochondria, and that's why first two steps occurs in the mitochondria of cell and remaining steps occur in the cytosol. First step is nothing but the synthesis of carbamyl phosphate. First step in the urea cycle is nothing but the synthesis of carbamyl phosphate. Okay, and it is a rate limiting step. Question इसके ऊपर आ सकता है, आ चुका है. Which one is a rate limiting step in the urea cycle? So remember, this one is a rate limiting step. Rate limiting step, irreversible step. इसे glycolysis में होता है आपका rate limiting step, irreversible step में देखा है. So likewise that in the urea cycle, so first step, क्या है first step? So this ammonia is converted into carbamyl phosphate. So first step, synthesis of carbamyl phosphate, which is a irreversible step and rate limiting step, or it is known as a rate determining step. Now in this step, 4 ATPs are consumed. Okay, I will see the urea cycle. I will explain you. This is a transaminase reaction. So transaminase is always coupled to, okay. So here is one amino acid and this is a keto acid. So ammonia group of this amino acid, ammonium group is transferred to keto acid, this keto acid, to form a new amino acid, and this skeleton is known as keto acid. Transamination reaction you can see over here. So this is a glutamic acid. ये glutamic acid है, उसका जो ammonia group है NH3 is a transferred. Now this is a pyruvate. This is a pyruvate. Pyruvate क्या है? Keto acid है. Keto acid. Right? Now ammonia NH3 group of this glutamate is transferred to a keto acid to form a alanine. Okay, and this glutamate without amino group is known as a alpha ketoglutarate. It will go into Krebs cycle. It is a very important intermediate. इसको ही फिर से अमोनिया अटैच हो सकता है, इसको ही फिर से अमोनिया अटैच होता है और बनता है ग्लूटामेट. सो दैट्स व्हाई ग्लूटामेट इज वेरी वेरी इंपॉर्टेंट है ना. सो इफ यू ईट नॉन वेज, अगर आपने 4-5 दिन कंटिन्यूसली या 8 दिन नॉन वेज खा लिया है ना, आप जानवर बन गए. एनिमल मूवी देख लिया आपने, एनिमल बन गया तो आपने, है ना. वो एक है ना, एक क्या बोलते चिकन लेग पीस वाला, वो तो जानवर कितना भी खाते हैं क्या मालूम. तो कहने का मतलब ये, इफ यू ईट नॉन वेज फॉर 3-4 days, so what will happen? So your urea synthesis will be increased, right, and your glutamate level of your body will be increased. So glutamate dehydrogenase enzyme will be increased, so that level is increased in the body, right. This is known as a transamination reaction or deamination reaction, right. Deamination is what? Deamination is removal of ammonia, right? Deamination that occurs in the liver. All other tissues can also cause deamination, but not of your glutamate. So oxidative deamination that occurs in the liver to form a alpha ketoglutarate, right? Liver. Now you can see this is known as a urea cycle. So first two test steps are there involved in the mitochondria. यहाँ पे इस यहाँ तक के स्टेप जो है माइटोकॉन्ड्रिया में होते हैं. Now glutamate mitochondria glutamate dehydrogenase, right? So in the leave mitochondria flavor glutamate dehydrogenase से क्या होगा? So glutamate dehydrogenase enzyme will convert glutamate or glutamine into alpha ketoglutarate, right? And it will release the ammonia NH4. Immediately this NH4, so that is converted into carbonyl phosphate. It is reacted with the carbon dioxide, two molecules of carbon dioxide, and it is converted into carbonyl phosphate by the enzyme carbonyl phosphate synthase 1. And this is the rate limiting step. यहाँ पे irreversible step है and rate limiting step. Important याद रखना है आपको irreversible step. Now this carbonyl phosphate combined with this ornithine. Ornithine is an intermediate amino acid in the ornithine is the intermediate amino acid. Now this carbonyl phosphate and ornithine they will combine together and to form a citrulline. This enzyme, this reaction is catalyzed by ornithine transcarboxylase. Ornithine transcarboxylase convert carbonyl phosphate ornithine into citrulline. Now citrulline में citrulline. Now one this amino group of this and one from this. Now it is NH2 C double bond NH, right? Now in this, okay, there is an addition of one aspartic acid. So citrulline is converted into arginosuccinate by the enzyme arginosuccinate synthase. Okay, citrulline is converted into arginosuccinate by the enzyme arginosuccinate synthase by taking this aspartate. Now you are going to get here two nitrogens, two amino group, right? These two amino group. Okay? Now arginosuccinase enzyme convert arginosuccinate into a arginine, and with the removal of fumaric acid, with the removal of fumarate, arginine amino acid जो था arginine. So from arginine is converted to ornithine by the enzyme arginase with the removal of urea. Urea is nothing but the NH2 C double bond O NH2. Now this urea is water soluble and it is easily excreted from your urine. Are you getting or not? समझ रहे हैं आपको यूरिया साइकिल है ना? नहीं तो गाना गाएंगे साइकिल साइकिल माझी सोने साइकिल, तो सोने साइकिल रोजापासून. Metabolic defect of urea cycle. So hyperammonemia type 1. It is due to defect in the carbonyl phosphate synthase जो rate limiting enzyme होता है. If there is defect, so there is accumulation of ammonia which is not converted, that is not converted into carbonyl phosphate. That's why ammonia will increase. So hyperammonemia type 1, that is due to defect in the carbonyl phosphate. Hyperammonemia type 2, that is due to defect in the ornithine transcarboxylase. Transcarbomylase. Ornithine transcarbomylase. Then citrullimia, arginosuccinate synthase is the defect. इसमें defect होगा तो citrullimia. Citrulline का लेवल ब्लड में बढ़ जाएगा. Arginosuccinic acid urea. So obviously due to defect in the enzyme arginosuccinase, so arginosuccinic acid urine में आ जाएगा. Hyperarginemia, arginine लेवल बढ़ जाएगा due to defect in the arginase. इसके ऊपर क्वेश्चन आते ही कौन सा एंजाइम का कहाँ पे डिफेक्ट. So I will save this recording once and then we will start again. So इसके बाद में अपन देखेंगे defects, because defects are very important. उसके ऊपर क्वेश्चन आता है.